experiment here. So the timer just went off, and now I'm adding 100 microliters of my catalase enzyme to each of the reactions. And I'm going to go in the same order in which I started the reactions. I'm reaching down to the bottom, pipetting up and down to add the catalase. Quick swirl. And then the catalase, if it finds any hydrogen peroxide, it will process that into molecules of oxygen and water. If you do have hydrogen peroxide in high amounts, you may see some bubbles in the reaction tubes. I don't expect to see any in these tubes because I have the no stress condition. There actually was no hydrogen peroxide added. There may be some hydrogen peroxide that the cells make on their own, but probably not enough to make bubbles in the reaction tubes. So now I'm ready to proceed with the dilution series. And I believe we've studied this now several times with count cells and also the mutate DNA experiment. There's some dilutions, but you want your setup to be very ergonomic, and you want to make sure your vortexer is close by. Have your pipette tip collecting. So begin with the null sample. Vortex before removing cells, fresh tip. Remove 100 microliters. And what I do is I move them to the side so I know that that's a tube that I've already processed. Vortex, change tips, remove, transfer. So these are the ones that are done, and these are the ones that still need to be completed. Continue with the Rec A treatment cell cultures. And now we're ready to seed the counting plates. I'm gonna start with the grid plates. Dilution steps 1, 2, 3, 4, and 5. 2 microliters on each of these spots, so dialing 2 microliters. So we're not going to use step 6, we'll just go for step 5. Rec A is the top row. Before removing cells, I vortex. Right away I see a problem that this tip doesn't reach all the way to the bottom of the tube. Okay, so I'm gonna have to use the P200 and set it to 2 microliters just to get it to reach. It's not going to be exactly 2 microliters, but it will do the job. So I'll start over here. 5, dilution step 5. So a little bit of liquid in that tip, and then I pipette, touch the surface. Keep going. And I'm just going to focus on the Rec A positive. There we begin again at step number 5 for the null. Okay. Spot grid plates are all set. Now we'll turn our attention to the counting plates. I'm going to do these in sets of 5, or in sets of 2. Dilution step 3, 4, dilution step 2. Okay, Rec A positive. 5, 4, 3, 2. So those are the Rec A positive and 6. And then nulls are here. 6, 5, 4, 3, and 2. So we can get this all in one view. Preloading the plates with glass beads. And I'll be adding 100 microliters of the diluted cells. And these are the null samples. So, and I'm going to be going from dilution step 6 all the way to dilution step 2. So 6. Step 5. Step 4. Step 3. It's off the screen, but I'm vortexing before removing each of these. And then we're going to do the Rec A counting plates as well. 6, 5, 4, 3, 2. Try to get the vortexer in view here. Yes, 100 microliters per plate. So Rec A, starting here with dilution 6, 5, 4, 3, 2. So 6, dilution step 6. Good. And then, like I've emphasized before, it's very important that the plates be dry before you put them into the incubator. So if you pre-dry the plates for 20 minutes, they should just soak up the liquid very quickly during the spreading process, and that's the case here. But if you see a wet curtain, you would want to air dry them further. The reason for this is that wet plates will allow the founder cell, the descendants of the founder cell, to swim away from the founder cell, and then you don't get a colony from each surviving cell. You get a lawn of bacteria because everybody's swimming to find new real estate. But if the plates are dry, then a founder cell will grow into a colony that's countable. So don't forget this critical detail. Make sure your plates are dry before putting them in the incubator. Stack them and keep them inverted. Again, this is to ensure that the surface of the plate stays dry. When they go into the incubator, they usually release a little bit of water as they warm up, and you want that condensation to fall into the lid where it does no harm. But if it falls on the media, if you forget this detail and leave them what I call sunny side up, then the media surface can get wet again, even though you put it in dry, and now the bacteria can start swimming. The bacteria are very good swimmers, and you have to have a semi-dry surface for them to grow on. Not completely dry, because then they can't get nutrients, but certainly not wet, because then they start to swim. I'm tidying up here, covering my ethanol. Getting some tape so I can secure the stack, both sides. And then you can label this security tape with your full name, so there's no ambiguity of whose plates these belong to. And then these go into the incubator for developing the founder cells into the surviving cells into colonies.